Imagine a world where a bacterium lives inside its host, infects the reproductive tissues, and transmits from a mother to all of her offspring. This bacterium is kind to its female host, inhabiting its cells without causing disease. But because it can only be transmitted through the mom, it adapts methods to prevent males from succeeding in the population. If you were a bacterium that wanted to suppress male success, what would you do? Perhaps you would kill the males, maybe feminize them so they can transmit you, or perhaps give the mothers the power to give virgin birth to all female offspring, making males obsolete. I'm sure you can imagine some of the ways that the world would change in response to the spreading infection. There is indeed a bacterium that does all of these things, and is also one of the most common animal-associated infections on Earth. My name is Dr. Dylan Shropshire, and my lab's primary goal is to understand how these bacteria hijack their host reproduction to favor their spread. You might be asking, why haven't I ever heard of this? Fortunately for us, these bacteria, called Wolbachia, do not infect humans or any other mammals. Instead, they live inside the cells of insects and a variety of other arthropods and nematodes. Here you can see Wolbachia beside the host Golgi and endoplasmic reticulum on the left, and throughout mosquito and wasp reproductive tissues on the right. In fact, over half of all insect species harbor Wolbachia in their reproductive tissues, and in many populations, they infect over 90% of individuals. And yes, Wolbachia do often kill males, feminize them, or give females the ability to give virgin birth. Though its most common method of manipulation is called cytoplasmic incompatibility, or CI. CI makes infected males, illustrated with filled sex symbols, conditionally sterile such that embryos die when they fertilize uninfected eggs, but survive if they fertilize infected eggs. This manipulation gives infected females a reproductive advantage that encourages Wolbachia's spread through a population. For example, here are the infection frequencies of two Wolbachia in eastern Australia. One does not cause CI, shown in blue, and the other does, shown in red. Researchers found that the CI-causing strain was relatively rare when they sampled in 2004, but when they sampled 8 years later, they found it had completely invaded the eastern seaboard. You might now be thinking, so it doesn't impact humans. Why should I care? Well, while Wolbachia do not directly infect humans, they are important to our health and that of our pets. For instance, have you or your family ever had a dog or cat with heartworms? Mosquitoes transmit these worms, a type of nematode, between infected hosts. Notably, these nematodes carry a type of Wolbachia that supplies them with nutrients they need to survive. As such, instead of treating your pet with antifilarial drugs, which often have severe side effects, your veterinarian may prescribe an antibiotic like doxycycline to kill the Wolbachia, and killing the Wolbachia causes the nematodes to die. In some parts of the world, it is also common for humans to acquire nematode infections that cause disease like lymphatic filariasis and river blindness. Here, Wolbachia are also the targets for treatment. Beyond nematode infections, Wolbachia are also proving to be a powerful tool to manage the spread of mosquito-borne diseases like dengue, Zika, yellow fever, and chikungunya. There are two methods currently in deployment, the first of which is called the incompatible insect technique and involves the release of Wolbachia-infected males into an otherwise uninfected population of mosquitoes. Because those males cause CI when they fertilize uninfected eggs, the offspring die. This can cause the population to crash. Several companies, including Google's sister company Verily, are using this method to reduce mosquito population sizes in California, Australia, and Southeast Asia. The second method is called the population replacement strategy. Like the incompatible insect technique, this method also leverages the conditional sterility of CI. However, it also makes use of another important Wolbachia trait, namely its ability to inhibit the replication of RNA viruses in its host. Since dengue, Zika, yellow fever, and chikungunya are RNA viruses, this means that mosquitoes that carry Wolbachia are less likely to carry these diseases and thus are less likely to transmit them to humans. To understand how this works, first imagine that you have a mosquito population with no Wolbachia. Now release Wolbachia-infected male and female mosquitoes into the population. Then, after reaching a certain infection frequency, stop releasing. Because CI helps Wolbachia to spread, soon nearly all individuals in the population will carry Wolbachia. Since Wolbachia-infected mosquitoes are resistant to RNA viruses, this new population will inhibit the spread of arboviruses to humans. In fact, the population replacement strategy has successfully reduced dengue burdens in release areas by over 85%, and the company at the vanguard of these efforts aims to protect 500 million people by 2030 using Wolbachia. So I hope I've convinced you that Wolbachia and CI are important. But what's our role in all of this? Well, my research is broadly focused on understanding CI's genetics, molecular basis, and evolution. As a PhD student, I contributed to the discovery of CI's genetic basis. Excitingly, these genes are not truly Wolbachia genes, but are instead encoded in Wolbachia's prophage, a virus that is integrated in the Wolbachia chromosome. In other words, we discovered that viral genes inside a bacteria are responsible for manipulating insect reproduction. Discovering these genes enabled a variety of other studies to investigate the biochemical and molecular basis of CI and investigate why CI sometimes causes 100% of eggs to die, while otherwise causing only small reductions in egg survival. These discoveries are important for explaining Wolbachia's success in disease control and why Wolbachia are the world's most common animal-associated infection. Using the fruit fly and its relatives as a model and tool, the Shropshire Lab will continue studying how Wolbachia's manipulations work, why they vary, and how they evolve. As a trainee in the lab, you would be an essential collaborator pushing the boundaries on our understanding of these traits that empower Wolbachia's wild success. As a first-generation high school graduate myself, I'm deeply committed to mentoring trainees and being their biggest advocate. In particular, I'm enthusiastic about helping trainees understand the unwritten curriculum of succeeding in academia, learn how to take a research project from conception to publication, and develop the skills they need to be successful in their unique career trajectory. Moreover, since the Shropshire Lab officially opens its doors in August 2023, new members will enjoy the benefits of working in a startup-like environment with brand new state-of-the-art equipment and facilities. If what I've talked about today is exciting to you, please reach out to discuss the possibility of working together.